We've had so many wonderfully talented people join us from the UNM Cancer Center, and today is no exception. I want you all to meet Dr. Jeremy Edwards. He's a researcher at the UNM Cancer Center. I want to get your title right. You are the New Mexico professor in the Department of Molecular Genetics and Microbiology, and in the Department of Chemical Engineering. You're a full member of the UNM Cancer Center.
That's correct.
Dr. Edwards, welcome.
Thank you for having me.
Good to have you. He just won a grant.
Yeah, can you tell us a little bit about that? And this was just announced as well.
Yes, it was just announced. Yeah, the grant is very exciting for us. The idea of our grant is to basically extend the capabilities that have been developing over the past decade for sequencing human genomes.
Wow.
And this is from the National Institutes of Health.
It is.
And you won this recently. It's a $1.35 million grant for a 3-year study. This is impressive. Congratulations, by the way.
Thank you so much.
So let's break it down to the basics. Human genome is what?
So the human genome is it's basically the set of DNA that's inside each and every cell within your body. Basically, it's generally thought that every cell has the exact same genome, and cells become different types of cells through different types of mechanisms, but the basic content of DNA is the same in every single cell within your body.
And tell us why it's important to know, to kind of break it down then, in regards to cancer.
So the DNA can be thought of as the blueprint to life. It contains the instructions that dictate what controls life. People have called it the language of life. I like to say it's kind of interesting. We'd always thought it was the language of life, but it turns out now that we can sequence genomes, it's a language we actually don't understand very well. Yeah. It's very complex, and that's kind of where our work comes in. So the cost of sequencing a human genome has dropped from around $3 billion when the Human Genome Project was completed. It took about 13 years and cost about $3 billion. Wow. And over the next decade, we've dropped the cost to almost $1,000, and it can be done in basically a week.
Wow.
So the cost and time for doing the work has dropped tremendously. But the problem is everybody's been working very hard to get the costs and the speed, cost down, speed up. And it turns out there's a lot of information we're missing. It turns out that when you sequence a genome, which is about, a genome consists of the DNA you get from your mother and your father. You get half of your chromosomes from your mom, half from your dad. And each set is about 3 billion bases, so the total is 6 billion. So 6 billion bases, and a base is just one of 4 letters, if you will, an A, T, C, or G. And to sequence that, you just determine the sequence of the A, T, C's, and G's. Now, it turns out sequencing is very complex. So you basically chop it up into little bitty pieces, put it back together like a jigsaw puzzle. And we have the help of the jigsaw puzzle box to help as we put this puzzle back together.
Right. We can see the picture.
You see the picture.
And know what we're trying to build here.
You got your little piece. You know what the problem with your picture is. There's many regions of the picture that are identical to other regions of the picture.
Ooh, it's a tougher jigsaw puzzle.
It's a very tough one. And it even makes it tougher because you have 2 identical jigsaw puzzles, one that came from your mom, one came from your dad, and they're almost identical. And this makes it very difficult. So today you can sequence a genome for a few $1,000s. The problem is lots of information is missed. And then that's where we come in. And that's what we're trying to do with our new grant, is basically provide more detailed information about the genome, how we can put this back together, and that might help fill in this gap between sequencing your genome and understanding what diseases one might be predisposed to, or if you have cancer, for example, what are the best or optimal treatments for an individual.
And that's good. I mean, that's especially good for cancer research, because that might be able to prove one day, well, this causes cancer, or okay, well, people are getting cancer because of genetics or such.
Exactly right. The problem is right now we
just
can
don't
sequence
know
a
right
genome,
now.
but it's really hard to interpret.
Yeah.
So there's tendencies that we're recognizing now, but we're not specific in saying that's going to cause cancer down the road. Is that what you're saying?
That's correct. Even some of the most common ones people hear about, the breast cancer gene, for example, that one 87% chance of developing breast cancer if you have that mutation in that gene.
And so there was Angelina Jolie, for example, recently had her genomes tested, or what's the sequenced? Yes. So how easy is it then for any regular person to go do that?
Okay. So the way I understand it, she actually didn't have her full genome sequence. She just had a specific region that was involved in breast cancer sequence, so not the whole thing.
Right.
So to get your own genome sequence, there's a couple ways one can do it. There's a program called the Personal Genome Project, or the PGP, and this is a project where one can actually have their own genome sequenced. You have to go through extensive examinations and testing and disclosures and so forth to make sure one is capable to actually understand this type of information, because it is quite complex. And there's the most common approach for actually sequencing one's genome, what people have heard of, that's on TV, there's commercials for them, and so forth. They actually don't sequence your whole genome. They basically determine a few specific sites within the genome that are known to exist in the population at different bases, different sequence at one specific site. So they'll sequence about 1 million of these out of the 3 to 6 billion bases. And then from these they can get some information about ancestry, or they might know that people who have this specific site at this specific position might be more predisposed to get a certain disease, but the predisposition might be 6% increase or something very, very small. Not a lot of definitive answers that can come out of that, but it's interesting.
Yeah.
I'm really curious about a lot of things here. I want to see if I can get some questions answered. So the information that's missing that you're studying and hopefully going to capture, what kind of info is that?
Right.
What do you speculate it will be?
So the genome is this very long string of individual characters.
Right.
And what one can obtain right now from the sequence is, at this specific position you should have an A, but you have a T. And these are called polymorphisms, or we call them SNPs, a single nucleotide polymorphism. At one spot it's different. And that's about all. You can find that and you can say you're heterozygous, meaning you have that on one of your 2 chromosomes, got from your mom versus your dad, or you're homozygous, meaning you got the same on both chromosomes. And that's about it. You can't do very well with respect to large-scale genome rearrangements. Especially in cancer, you can have this chunk of the genome stuck over on this part of the genome. That's not supposed to happen. With the sequencing technologies today, those are difficult to find. You can have deletions, you can have amplifications of the genome, you can have all these different types of things happening, and with the technologies that exist today, those are very difficult to find. It's also very difficult to find connectivity along the genome. So I said you have 2 sets of chromosomes, one from mom, one from dad. If you find some mutations or polymorphisms or what, you can't place them on one chromosome versus the other relative to one another. And it turns out that's probably very important in many diseases. Basically, what is connected to what can impact the disease that one would get. And these are completely missed with existing technologies, and so that's what we want to do, is try to capture a more complete genome rather than just so take the genome and actually sequence it, rather than take the genome and put together a table of single nucleotide changes that might exist.
So if I understand correctly then, if people do have their genome sequenced for some genetic problems or whatever, that doesn't guarantee them. So if I go and get my genome sequenced and it says, okay, well, you're clear for everything that we tested, that doesn't mean I'm not going to get cancer 10 years down the road, because it hasn't. It hasn't done everything. It's not, you're not able to detect everything. Right?
Not able to detect everything, and even everything you detect may not have come up in the assay. It is, the way we sequence a genome is you take it, you chop it in little bitty pieces, and you just randomly sequence it. By random chance, you will miss certain spots within the genome. And another example, it is very complex. Even if you don't have the breast cancer gene, it doesn't mean you're not going to get breast cancer, because that gene only accounts for about 5% of all breast cancers. So it's a very complex picture, and so there's lots more to understand, and that's what we hope we can
Yeah, we could have you on all day asking questions.
What you're hoping to do is paint a clearer picture when people get their genome sequenced. This is fascinating work. Boy,
Thank you.
This
when
is
you go to the
amazing.
office, your world is amazing. I love it. It is really interesting to meet you, and I have so many more questions.
I know.
and we're out of time. I really do. This is a fascinating area of research.
Well, it's a requirement to be faculty members to be able to talk for extraordinarily long periods of time and say almost nothing.
Well, no, you're saying a lot, and
Yeah.
and I want to talk to you again, so we'll invite you back.